man, he's got to start over. And I felt bad because I wasn't even thinking about it.
Yeah. No, I did it. So it's kind of one of those things that, you know, even now I was like, oh, I got to start recording. I'll reset it so that once I turn it on, I record. That probably be
Yeah, no worries. You're rocking and rolling this morning. That's why I was just kind of like, oh, I felt bad, you know.
Yeah, that's all right. All right, guys, got about another 8 minutes before we start up. Bunch of all, good to see you there. Hope things are going well with everyone. Sorry we weren't able to have lab today. We have an exam coming up. Next week. It's online.
Morning, guys. Morning, Professor.
Good morning.
Professor, when I was watching
I'm sorry, you're kind of fading in and out.
the module 5, it's like the audio.
So, Amy, I did not get any of that. Can you try again or put it in the chat? And you're frozen. Okay, there you are. You look like you're working now.
Oh, can you hear me now?
Yes.
Okay, so your second video in the module 5, it's like you're recording it like twice. So there's already a recording, then you're explaining it again. So it kind of overlap. I don't know if you check that one.
Okay, I'll have to take a look at that.
Okay, thank you.
That's for the Zoom lab?
I think so. That's about the cell, parts of the cell.
Okay, let me go look at it. We got about 5 minutes before class starts, everyone, so bear with me. So Emily, I'm going to go down and look at it. And so you're saying you're looking at module 5. I have the slide decks, and you're saying the cells, you get parts of the cell. You're saying you look at this one and it's looking a little weird.
Yeah, the second video.
Hello, and welcome to Biology 240. This is microbiology, and this is microbiology introduction to about cell. And we're covering. And remember, these lectures are meant to supplement and highlight what you use in the book. And then during.
All right, I'll have to change that one.
Thank you.
But thanks for letting me know about that.
So I have a silly question, Dr. Cap. This review for the exam, is it going to actually be like these questions or just similar to this format? Let me ask you that.
Similar to that format.
Got it. All right.
Because again, you guys could look it up. If I gave you that same exact question, I'm testing how quickly you can cut and paste that from there. And that's not the intent.
Hey, Dr. Cap, where's
Yes,
the
sir,
study
Michael.
guide located again, please?
Where the what?
Like the study sheet or the review sheet?
I put the review in chat, but review, I'll go over today. You should be able to see the review on the modules, the last 2 modules. I put the review in there.
Okay, thank you.
But if you... I just put it again in the chat. All right,
Okay.
guys, got about 2 more minutes before we go on, and I'll do a kind of a little bit of a demo. So like always, I post a lecture. Again, we're going on chapter 2 and 3. Chapter 2 is looking a little bit more on the prokaryotic diversity, and chapter 3 is looking at cells, specifically the prokaryotic cells and eukaryotic cells. I did a lecture on that. Emily just is always really good at pointing out my faults. Told me that the lecture on that one has like a double lecture. So I'll go back and hopefully pull out that other lecture on there, or I got a lecture on it, it's where it's timed. So I will kind of fix that.
How many questions are on the exam, Dr. Cap?
I haven't made it yet, but typically I do about, I'll probably have about 30 multiple choice or true false. I'm scheduling you to take about 30 minutes to do that. I'll probably have 10 or 15 short answers. Again, scheduling you about 30 minutes to do that. And then I will have, typically I have an essay question. And I schedule about 30 minutes for you to do that as well. For me, multiple choice, especially with you guys being able to look at stuff online and stuff like that, multiple choice for me is kind of like a giveaway. Those are meant to be a little bit easier. I try and make you think, but there's only a certain number of ways I can make you think on a multiple choice. For the short answers and the essays, you will get stuff that will not, you shouldn't be able to look it up easily. So it's something that you need to kind of think about. It's something that you need to assimilate information. So again, you need practice on that. Anyone have any questions? So we now have 38 students in here. Thank you for coming and spending some time with me. I will be lecturing in a second, but before I start up, does anyone have any particular questions? Remember to sign in on the Google chat. That's one of the ways that I have of taking roll in class. We have exam one. Exam 1 is 1 week from today, the 21st of September. Okay. I'm hoping a little bit more Wednesday will be a little bit more of some kind of review material on that, although I'll go over that a little bit today. Before I start, does anyone have any questions, comments, concerns? Amy already told me that the the lecture for chapter number 3 is kind of messed up. I think I recorded 2 lectures at the same time on there, so they're a little bit off, so it's a little bit hard to understand. I will go through and try and fix that this afternoon and put that up. What I'm going to do is I'm going to share my screen now, and I'm going to go back. So I was making some edits. I'm going to go back and show you a little bit of things that you should be able to see.
I have one more question, Dr. Cap.
Mm-hmm.
When we take the exam, let's say, for instance, we don't know the answer to a question and we decide to skip it and work through the exam, will we be able to come back to that question or no?
Yeah, you should be able to do that.
Okay.
That's my intent anyways.
Okay,
All right,
thank you.
so I'm going to pop this on home. Student view. All right. So if you go to home, we have our recent announcements. We have things that you need to know about Skyline, general microbiology. And so I put things in reverse order. So here's week 5. If you click week 5, we have our lab materials that are in there. Wednesdays, students, you will have to know, we'll have to figure it out. If air quality is good, I think it's predicted to be good. You will be coming in and doing your validation. For Monday individuals, we're going to be pushing your validations back a little bit. So we're not going to be in sync, but that's okay. If we click on week number 5, ideally what we would see is for all, again, we look at, I produce a slide deck of slides that I will be using. That's basically from the figures from your text. Chapter number 2 and chapter number 3 is kind of what the chapters we are going to be looking at. That is also on the course syllabus. I give you a lecture, a video of the lecture. I go pretty fast. You could slow that down or you could speed that up if I'm too slow. A student did tell me, so lecture number 2 is there. A student did tell me that the link for number 3, it's actually it has 2 lectures going on at the same time. And so I will fix that, so hold off a little bit on that. But again, if you see that, kind of let me know right away. This course is being recorded, right? Yeah,
Yes.
so we are being recorded, so let me go. Also realize that when I am giving a demo up here that I can't see the chat. So if you have a particular question, you can shout up. Or if somebody reads something that looks like it should be brought to my attention, please have it come up to my attention. Typically, I will post the Zoom lectures for the class. So this is for Monday. And then what we're going to do is we're going to do a short discussion today. What I wanted to do is go over some of this material. I have a shorter slide deck, or what I call a slide deck review. So I go over the quizzes and kind of see things that people have gotten wrong or things that historically people have missed, and I'll go over those a little bit in class. And then we are also going to have an in-class assignment. That should be here, this multimedia group assignment. So I want to lecture till roughly about 12 o'clock, and then I'll give you that assignment. And that's not on there. That actually should be popping up there already. And I'll give that to you in a second. On Wednesday, I will record Wednesday's class. I'll probably have another slide deck on there. I want Wednesday a lot to be a little bit more of a review or questions for the exam. So there's some quizzes that you need to take. Quiz number 4, which is on chapter number 2. Quiz number 5 was on chapter number 3. Quiz number 6 is kind of all the review materials. I would make it like a practice exam. And so what you want to do is you want to do as much studying as you can, and then you take quiz number 6. And that should tell you about how you're going to do on the multiple choice question of the exam. Again, it works a lot better if you do it without looking stuff up on Google, but you already know what I feel about that. The materials, exam number 1 for this should be the materials for exam number 1 are posted here. There's a page up that you can take a look at it on here, and so it's on here as well. In order to study for the exam, like is like. So for multiple choice, what I would say is you need to take the quizzes and see what you're doing on the quizzes. And so what I would suggest is that you take the quiz without looking stuff up. Because the quizzes, I take your highest score no matter what. And so I have no problem with you taking the quiz, but what you want to do is take a quiz, and if you get a 0 on it, then that quiz, you have to study that material more. If you're getting a 10 out of 10 on the quizzes, then you know what, maybe look at another chapter that you don't know as well. The other part of the exam is going to be short answer and kind of an essay. And how you can study for those is if you go to our discussions. If you go on the discussion, I have a discussion labeled exam number 1, materials on exam number 2. I need to change that. I'm sorry about that. So exam number 1. So if we click on this, looks like we have some people in here. So if we click on this, hi all. So it says, I'm going to read it off. Hi all, this is a discussion. I will be posting possible questions or ideas that you will see during your first exam. Please ask and answer the following questions. I ask that, some of you are really rigorous students and you're going to go through and you're going to answer all of these. I don't necessarily want this to be a discussion between me and the first student who is able to answer all of these. So answer some of them. If you can answer some of the other ones, put it down and hold off on it a little bit and then see if your answer is right. Don't necessarily write everything down. So I may ask, if I'm talking to it's not intended for me to talk to 2 or 3 students, although I love talking to students in here. If I have one student who's answering everything, I may ask you to slow down a little bit or kind of save your answers. And so I asked a question, can you write out and describe a situation where you used a scientific method? So again, a typical essay question, what I may have you do is maybe pick something from your life and show how you can use the scientific method, explaining the parts of the scientific method as you go. And so this individual, Amy, actually did it for baking a loaf of bread or a loaf of bread that did not bake. I especially liked it because it is microbiology related. So it's a really kind of good example. And we'll look at that. And if we go down a little bit, so again, here's another possible question I can have. I'm not guaranteeing that these questions would be on an exam, but this is a little bit of how I want you to think about it. Give an example of your favorite microorganism. Why is it considered a microorganism? Explain how it is related to all other microorganisms. Does it cause any diseases? If so, how can we cure or prevent the disease from occurring? How is it like E. coli? And so you're going to take your typical gram-negative proteobacteria and compare it to it. So I have a number of these that are up there. Okay, so go ahead and try those, and I will be looking at these and monitoring those. And what I do is when you answer the question, I will try and tell you exactly what do I look at when I go through and I look at exam questions. So I will be looking at that. Is there any questions on this? No, I don't see any.
The whole second part, you were breaking up and your screen froze.
Oh, sorry. So so these discussions are going to be what the short answers or the essays, how they are going to look like on the exam. So if you want practice for that, please go ahead and take a look at these. Because again, my constraints is that you guys have the internet at your access. So I'm not going to ask you to define a microorganism. I'm not going to ask you to a number of those different things. But what I will ask you to do is to take knowledge that's generally available and synthesize something new from it. And so again, we're looking at kind of these kind of higher orders of thought and education, which my colleagues tell me is something that we should have been doing anyways. Any other questions on that? Okay. So I'm going to go back to the modules. And
Professor?
yes, ma'am.
Oh, did you hear me?
Can you repeat what you were saying?
Yeah, we just lost connection about or at least for myself, you froze for about 20 seconds or 15 to 20 seconds or so.
Yeah, same, Doc. I can't hear you at all.
You can't hear me at all?
You freeze and break up as soon as you scroll down to the second part. You keep freezing and break it up. Your connection's like getting lost.
Okay.
It's the whole app.
It's the whole app. All right, so let's do this. So just look at the discussion board. Has everyone got that?
Got it.
You know,
I have a question, Dr. Cap.
Yes, ma'am. Who's that? Is that Alexandria?
It's Katrina.
Hi, Katrina.
Since lab is canceled for Monday already, and if Wednesday gets canceled, will you revise the study guide on gram staining, or do you still want us to just look up that information?
Nope. I want you to look at gram staining. It's an important part of it. So,
okay.
And again, you guys all made you guys done most of the lab. It's a small part that's left for you to actually come in. So you guys have been working on the gram staining material for a while. So,
okay.
So that is still there. And I'm hoping that the Wednesdays people, we will, it looks like things are cleaning up, and I'm on campus and it doesn't seem too bad. I'm hoping they do open it up for tomorrow. Okay. All right, any other questions? So sorry, it's going out, but what I want you guys to do is to look at the discussion board, okay? Participate in the discussion board. The next thing I wanted to do is to go, so I'm going on to week 5, and I'm going to click on the slide deck review. All right, so it's going to pop up. I'm going to do present. So I'm going to try and go steady and slow. For week 5, module 5, we were really looking at chapters 2 and chapter 3. We're looking at so bacteria, characteristics of bacteria, how we look at bacteria, and chapter number 3 is looking at kind of cells and kind of what cells do. And we'll have an activity today that will hopefully help with that. Again, in looking at the quiz, the average for the quiz was about an 80%. It took you guys about 8.2 minutes to kind of get through the quiz for chapter number 2. So it looks like the people who have done the quiz have done pretty well on them. But I think some of that is you guys are looking, you guys are looking stuff up as you do it, which is okay. Chapter number 2 is meant to be an introduction into microorganisms. And when we look at a microorganism, we're looking at things that are under 0.1 millimeter at some point in their life cycle. And so does anyone remember what's the significance of 0.1 millimeter?
Something that we cannot see with the naked eye.
So something that can be viewed with the naked eye.
No, we cannot. We cannot.
Something that's under, why not?
Because they're very, very, very tiny, so we can use a microscope.
Okay, remember this is a science class and you're not presidents of the United States, so you can't say very, very, very tiny. What specific language do I want to hear?
Microscopic.
No!
The eye is unable to see less than 10 micrometers?
No. You got 10 wrong. 0.1 is 100 micrometers.
0.1.
So microorganisms are 0.1 millimeters, so we need to use
Okay, but you need to know what the difference between magnification and resolution is. And so what we're looking at, the resolution of the human eye is roughly at about 0.1 millimeters. And so resolution is the ability to see or to isolate two different objects in an image. And so the better the resolution that you have, the smaller these things can be together that you can see the difference from them. Do you guys see what I mean there?
Doc,
Yes?
can I clarify with the magnification resolution? So is it meaning like magnification can only be useful with enlarging images, but without resolution, it'll be meaningless?
Yes.
Okay.
So that's one of the reasons why you can buy, a really cheap microscope that has 1000x magnification, but you still can't see anything in it because it has poor resolution.
Like the fold scope?
We got to look at it a little bit more. Supposedly the photoscope has some fair resolution.
I think it was like, when I did my study guidance, I think it was like 40 only or something, or 100x.
That should be able to, yeah, you should be able to look at something 100x.
2 microns.
Yeah, we'll talk about it a little bit later, but yeah. But again, part of this is when you describe stuff. So when your essay questions or your short questions, I'm looking for this terminology. So to say you can't see things that are really small, you need to tell me why. Okay, so that question, that answer is correct. But again, remember, I'm testing your knowledge of microbiology. So you need to use the language and the jargon of microbiology. Okay, how do we view microorganisms? So I like Gary Larson is really kind of good at this one here. And so here is an early microscope that has very poor resolution. And the caveman is sitting on top of there saying, it's a mammoth. And here we have early microbiologists looking at Petri dishes. So one of the things about magnification is it makes things appear closer. So I guess the closer that you get to the Petri dish, the bigger things will look. But again, that's still pretty hard. What this is showing you is kind of the two different forms of light, what we call light microscopes. We have what's called a bright field microscope and a dark field microscope. And so just like what it says, in the bright field microscope, you have this bright background and your images appear dark on that bright background. And so what's happening is you have light shining from underneath your image, and it goes through the image. And when that light gets absorbed, refracted, or reflected, it changes it so that you can see the image. For a dark field microscope, what you're actually doing is you're shooting light on the side of an object, and then you're looking at that light as it reflects off. So you can't see that light in the background. It's dark because you don't have light going there. And if there was nothing there, the light would just move across your field and you wouldn't see anything. And so that's a different type of special microscope. You should be aware of kind of what refraction and reflection is, and what's going on with a refracted reflected ray is when light actually bounces off or is reflected by a surface. So a mirror is very reflective. So when you look in the mirror, the light is going into the mirror and it's reflecting back into you, and that's how you see it. And refraction, what we're looking at is when light goes from one media to another, it kind of bends a little bit. I'm not going to get into the physics of it, but it bends a little. It bends a little bit. And so what we see is, and that should be the next slide. Actually, well, so when light goes from one media to another, we have this slight bend. So sometimes we can see that in glass, or what a lot of times you see it is if you have a glass of water and you put a pencil in the glass of water and it looks like the pencil is bent. That's due to that refraction. And we are going to need to understand refraction to look a little bit at our microscopes, and I'll give you that in a second. You guys should know what the parts of a bright field microscope and kind of what they do. You should be able to follow light through a bright field microscope. And so can anyone shout out, where do we put our eyes? Where are we going to look at an image?
An ocular lens.
An ocular lens over here, okay? And so what's generating the light?
The illuminator.
Number 7, illuminator. And so you should know that the light gets generated here. It's going to go through a condenser, through the diaphragm, through your ocular, and then through some prisms and back into your eye. And you need to know a little bit about how that happens and what's going on there. Oil immersion. So this is a picture of an oil immersion lens above a slide, although there's a cover slip here. When you guys look at your bacteria that are heat fixed onto slides, the great thing about them is we don't need to use a cover slip. This cover slip actually can be too thick sometimes. So what you see, there's some oil in between here. And so normally what happens is when light goes through your slide and then your sample, when it hits air, it's going to refract, right? So it bends a little bit. What that does is that will affect your resolution. Things will look kind of blurry. You know, it's hard to tell two different points apart. So what you can do is you can put a little bit of what we call immersion oil in between there, and that immersion oil has the same refractive index as glass does. And so what happens when light goes through, it goes through your image and it goes through the oil, it won't refract, it won't bend. And so that way the image looks actually quite a bit sharper. So when you're looking at something underneath your 100x objective and it's blurry, it could be that your immersion oil is not there. And so if you put your immersion oil drop in the wrong spot, or if you moved it a lot, that immersion oil won't be there. Sometimes immersion oil could kind of go bad, get cloudy, or not be washed off. And so again, when you're looking at it and it's not in focus, there's a number of different things that you can look at. Does everyone get that? Yes?
So will leftover immersion oil affect the way you look at a slide that doesn't need immersion oil? Because like,
Yes, it will make it blurry.
okay, yeah, because like the last on our validation last time, like I noticed that there, some of the objective lenses had oil, so I had to keep wiping it off because it was like blurry.
Yeah. So that's one of the reasons why when you get your microscope come in, the first thing, you know, and nowadays with COVID, and you should do this anyways, but you should always use disinfectant and sanitize your work area and sanitize everything that you're working with that's not yours or that could possibly have been used by somebody else. And so let's just say from now on, and it's good. We should have been doing it anyways, but in times of COVID especially, make sure that you wipe everything down. And that's really, it's good. It's good practice to wipe off. You know, I'm sorry about it, but sometimes other people use the microscopes and they don't clean stuff. And so if you wipe,
It's good.
Yeah, but it's kind of like water being put on something. You're looking at it and you're looking through water, and it just makes it difficult to see.
Okay, thanks, Doc.
Okay, no problem. What kind of sandwich you got there? You're on mute.
Turkey and cheese and some Doritos.
In the sandwich?
Oh no, Doritos are on the side.
Oh, okay. All right. Good.
All right.
All right. So again, a lot of us are clinicians, and we want to know the microorganisms that we're looking at. And so one of the first questions that we are going to ask our microbes once you get a microbe to deal with, is it gram positive or is it gram negative? And so what your first question, you ask a microbe. Again, you can't ask the microbe directly, so what you're going to do is you're going to do a differential staining technique on it to take a look at it. And so here are all the steps, and a lot of you guys have repeated these steps for me. You should have read an SOP for today's laboratory or Wednesday's laboratory. And so an example of a gram positive, so here's what's going to happen. So here's a gram staining process. I want you to be aware of this process and to be able to talk about it. And so this is a gram positive cocci, and this is a gram negative rod. So when we stain it with crystal violet will actually stain cells kind of a purple bluish color. And it sticks to all cells. And basically, it kind of sticks to the peptidoglycan. We'll then wash off the excess stain. You then put a mordant in there. And what the mordant does is it actually binds to crystal violet. And so crystal violet does stain these cells, but the crystal violet stain is pretty weak. It doesn't bind to the cell very easily. And so if you take a little bit of, here's your D-stain, and 95% ethyl alcohol, and if you put a little bit of that on there, it would wash it out. The trouble is if you take iodine, the iodine will bind to the crystal violet and make it a little bit bigger. So imagine, if you will, I have, so I'm showing you, I don't know if you can see it, but I have one of the test tube holders from the micro class up in here. And so if I put a, and so I'm putting my little lens in here. If I put a sample in here, it can't get out. And so what happens is when you add iodine, the iodine actually is a mordant. It changes the size of the crystal violet, so it's harder for the crystal violet to get out. And so what happens is when you decolorize, and again, between each of these, when you decolorize, what happens is your gram positives that have lots of peptidoglycan, they don't decolorize. But again, if you decolorize this a lot, they will decolorize. So that's one of the reasons why it was really critical. A step that was really critical on this is that you don't decolorize for more than, it said between 5 and 10 seconds. And I was thinking more of like 5 seconds. You decolorize it for 5 seconds, and then you quick right away do a rinse. What's going to happen there is that's going to decolorize your gram negative. That is now relatively clear. It's not going to decolorize your gram positive. You then need to add what's called a counter stain. And so we use safranin, which is kind of a red or pink color. And what we see is that even though the gram bacteria, the gram positive bacteria will stain with the safranin, because you have a dark blue stain there already, you can't see the light pink stain. However, your gram negatives will actually stain that kind of light pink. So when you look at it underneath the microscope, and this is what you guys should see on Wednesday, and then 2 weeks from today on Monday, is here we have some gram-positive rods because they're this bluish color, and here we have some gram-negative, actually Vibrio. And so again, and actually this looks like we have some gram-positive cocci in here as well. And so again, it's kind of a judgment call when you look at these things underneath the microscope. If I had a whole bunch, so here we have these different shaped rods, that's probably a different species. They're a slightly different color. Here we have these kind of gram negatives in here, and so that's the ones that we are looking for. Again, you need to be able to understand when your gram staining didn't go correctly, and I'll give some examples of that.
Professor, I have a question.
Yes, ma'am.
Is it because the gram negative, they colorize because they have a thin peptidoglycan, or?
They have relatively a smaller portion of peptidoglycan, and I will, I have another slide that we kind of go through that.
Okay.
Yes. The second thing I want to do here is that for 100 years, asking bacteria whether they're gram-positive or gram-negative have actually worked pretty good for us. But trouble is we're finding more and more bacteria, and we have to have more and more ways of identifying how they are different. And so a modern way to ask microbes if they have a particular structure or something is actually what we call immunofluorescent staining. And so we can take an antibody and we can bind it to a certain molecule. Let's say it's a certain molecule that happens on T. pallidum, which actually causes syphilis. What we can do then is we can actually stain that molecule with other antibodies that are connected to what we call a fluorochrome that will turn a different color. And so nowadays we have a lot of different ways of actually looking at microorganisms underneath the microscope and telling kind of what parts are what. And so if you look at it here, this is a picture of a cheek cell from a human being where the nucleus, the DNA, is stained with a blue dye, and then some of your microfilaments are stained with a green fluorescent color and a red fluorescent color. I think this is the different stains. And so we can get an idea of what a cell actually looks like. And so underneath the microscope, we really couldn't see anything. But here we can look at specific things. I'm not going to get into this, but there's 3 individuals who got a Nobel Prize for this earlier on in this century. Martin Kalifi and Usama Simimura, the Japanese have been really good at that, but I think they were in the United States when they did that. So I think what I'm trying to get there is, where science really works is where we can take a relatively complex issue and try and make it as simple as possible. And so again, what those individuals won a Nobel Prize for is we had this problem. How do I know what I'm looking at underneath the microscope? Well, you could take an antibody and bind it. That antibody will only bind to a specific protein or antigen, one specific one, and I can tell where it's at underneath the microscope. And we tend to see where science kind of goes forward is where we're able to take relatively complex issues and make them a little bit simpler for all of us to understand. And so, again, Louis Pasteur had this, again, when he was looking at this thing that we call spontaneous generation, and the spontaneous generation is one of those kind of ideas that just won't die. I mean, intuitively, it kind of makes sense. Oh, these things appear out of nowhere. It's magic or whatever. We all want to believe in magic and stuff like that. And so what people are saying is they proved, hey, cows just don't appear out of nowhere. Horses, big things don't appear out of nowhere. But people still said, oh, well, viruses and bacteria can appear out of nowhere. And so what he did is he showed that you can take a little bit of nutrient broth, put it in a flask, bend the top so that gases can go in and out, but microorganisms can't go in and out. And he shows that this can remain sterile. If you break off the top and expose it to air with bacteria in it, then the bacteria will grow. So it's not that this media is there. And so what we see is he took some, people had kind of thought that disprove spontaneous generation for microorganisms. But Pasteur, doing this relatively simple experiment, said, you know what, no, it doesn't happen. Why does this have to happen? Because think about it. If you're going to cure a disease, if a disease can just spontaneously pop out of nowhere, then you're not going to be able to cure diseases. Okay, people are always going to get these weird diseases. Yet, if you can show that you have to have exposure to microorganisms in order to get the disease, even if you don't have a cure for the disease, if you prevent people's exposure to the disease, it's not going to happen. And so what we see, a lot of our cure, or a lot of the reasons why we don't see as much disease nowadays, is because we know how diseases spread. And so even now for COVID, we don't have a cure for COVID. We don't have a vaccine for COVID. Yet you could prevent getting COVID by wearing masks, sheltering in place, sanitizing your area, etc. These are all things that we can do and that we should do because they're relatively cheap. Another idea that came that we're pretty sure of now, but actually this was developed in 1920s by Walden, was something that we call the endosymbiotic theory. And in the endosymbiotic theory, what we see is that when we saw the evolution of microorganisms, the eukaryotic organisms are pretty complex. And we think why they are so complex is because basically what happened is that these larger organisms engulf these smaller organisms, and rather than killing them and breaking apart and eating them and consuming them, what they did is they formed a symbiotic relationship, or they enslaved them. And so what they did is they had these bacteria come in that were really good at making ATP. So if you give them sugar, they're really good and very efficient at making ATP. And so those are the mitochondria. Chlorophyll and the photosynthetic evolved from kind of chloroplast, actually evolved from blue-green algae that were taken up. And in the 1930s, we didn't, while somebody said, hey, this is a really good idea, let's do it, they laughed them out, they laughed them out of the university, that can never happen. But what we're seeing now is later on, with better techniques, we're able to see that when we look at the proteins, and actually chloroplasts and mitochondria actually have their own little genomes. They actually make their own proteins. They have their own DNA. And when we look at that, we can see that those organisms are very related to true bacteria, the eubacteria. And so here's a case where something happened. You guys need to know the parts of a prokaryote. So here's a slide from your book. We'll go over on that. We need to know how these parts work. And so what I'm going to look at here, what I'm going to tell you this is, what you may say, why is a cell wall useful, is that we have something that's called a plasma membrane. The plasma membrane controls everything that's on the inside of the plasma membrane is a part of life. Everything that's outside of the plasma membrane is kind of other. That's not part of life. The plasma membrane controls what materials come in and out of the cell. Trouble is in that plasma membrane, so what we tend to see is that chemicals move down what's called a concentration gradient. And so if we have inside the cell, we have a 80% water, 20% solution. On the outside, this is what we call isotonic, when we have a 20% solute solution, 80% water. So what's going to happen is the water is going to move out in equal amounts that it moves in. And so these plasma membranes don't necessarily totally control water. Water can move freely across a plasma membrane through lots of plasma membranes. And what happens is that if inside of a cell we have a 20% solution, 80% water, and outside we have 40% solution, 60% water, what's going to happen is the water is going to want to go out and diffuse. The molecules can't come in, they can't move down a concentration gradient so that the water is going to move out of the cell, and that's a process that we call osmosis. And the opposite happens. When inside the cell we have a 10% solution, 90% water, on the outside we have a 20% solution, we see, on the inside of the cell we have a 20% solution, what's going to happen is that water is going to move into the cell, and it fills up the cells. And what will happen is that cell will actually burst, or what we call lyse. So it's really critical that we have a cell wall around these cells so that they don't burst. Human beings don't have cell walls. So if you get I don't know if you've ever gone to the hospital and you've gotten an IV solution. If you look at that IV solution, it's going to be what we call an isotonic solution, either a saline or a sugar solution. Because if you gave somebody just pure water, it wouldn't be good for them. Here's looking at the plasma membrane. You should know what a plasma membrane looks like and what parts of it kind of do. I'm going to give you an assignment, and we're going to look at it, and we'll review it a little bit. But basically, the plasma membrane is made out of phospholipid bilayer. And so we have hydrophobic and hydrophilic regions of this membrane. It's a lot like a soap bubble. And inside this membrane, we have various proteins that are embedded in it. So it's a lot like a mosaic. And a mosaic is basically, I don't know if you've ever seen where you take tiles and you can make an image. You have different tiles that are different shapes and colors. You can put them all together. And that's exactly what this plasma membrane is. But again, it can bend and it can move. And so for a lot of times, smaller molecules, especially smaller molecules with no charge, can move across a plasma membrane. How we can do what's called active transport, you can actually have what's called a transport protein that recognizes. So in this one here, this transport protein is going to recognize the sugar. It moves the sugar across. And what happens is these things are pumped by the hydrolysis of ATP. And so this activity of ATP moves the sugar across. And so what we're seeing is that all living cells actually are utilizing ATP to move substances across the cell when we see that. How are you guys doing? Okay, I'm throwing a lot of stuff out there. So this is looking at the peptidoglycan. And so the peptidoglycan is alternating residues of N-acetylglucosamine and N-acetylmuramic acid, and that these are bound together by basically a pentapeptide, right? So that's 10 amino acids together. And so this gives us a very strong structure. It's kind of like the filaments inside the mask. I'm not going to ask you exactly what is going on here. And actually, if you look at it, the structure of both the gram-positive and gram-negative peptidoglycan is basically the same. It's a little bit different in their peptide. But this is the main figure that I want you to look at. And so what we see, how this arrangement is in the cell wall and the plasma membrane arrangement is, in the gram-positives, we have your plasma membrane, and then you have lots of peptidoglycan cell wall. In the gram-negatives, you actually have two membranes. You have an outer membrane and an inner membrane. And in between this inner membrane and what we call this periplasmic space, this space in between the membranes, we have a little layer of peptidoglycan. And so when you do a gram stain, you're actually flooding this peptidoglycan with crystal violet. You're changing the size of the crystal violet so that it hangs out there a little bit more. But when you flood it with alcohol, this small, relatively small section of peptidoglycan washes out. Whereas here in this gram-positives, you may wash out the crystal violet from the first, second, third layer, but you have a fourth, fifth, sixth, seventh layer that are in there that remained stained. The second thing that we have on here is on your gram-negatives, you tend to have something that's called a lipopolysaccharide that's kind of hanging off the outside of the membrane. And that's what we call lipid A, or kind of the O antigen. So your gram-negatives have these kind of outer cell surface that looks a little bit fuzzy, and it's very antigenic. And so an example of that O antigen, so here is the phospholipid, and then here's the core of the A antigen, the lipid A antigen, and here's this O antigen that's on it. The reason is with this is that you may look at, whenever you look at something in a hospital, an IV bag or anything, it's actually, these are meant to be what they call depyrogenated. And a pyrogen is something that actually causes a fever. And so what happens is you can sterilize something, but if it was contaminated with E. coli, those pyrogens are still left. That LPS, the lipopolysaccharide, is still left. And people will respond to that lipopolysaccharide. It causes fever, it causes swelling and stuff like that. So even though they don't have an active infection, they will be reacting to materials. So it's very critical that materials, bandages, and catheters and things like that that are used in the medical setting are depyrogenated. This last one here shows you kind of what a eukaryotic cell looks like. There's a number of different parts, and these all do kind of different things. I wanted to kind of give you a little bit of time for the last half hour of class. I'm going to give you an activity, and I'm going to split you up into groups. Eukaryotes are a little bit different in that, again, eukaryotes, to eukaryotes to be part of microbiology, at some point in their life, they have to be less than 0.1 millimeter or 0.2 millimeter. They have to be really small and hard to see with the naked eye. They can still be multicellular. And so, again, they can be multicellular, but they can still be really small. Okay, the cells that make them up are still really small. So what I'm trying to do here is I'm going to give you an activity to help you kind of visualize or learn something. And so for me, a cell is a lot like a really high-tech factory. So the type of factory that we're going to need when we go to outer space or when we go to Mars. And so if you look at a factory in this diagram that I'm showing you here, this shows you a factory of that you have input coming on in this number one. Here you have a guy that puts it on a conveyor belt, and you're going to have things modified on this conveyor belt. Once things are modified on conveyor belts, you have another woman over here, and she's going to put it on different belts to be modified, possibly different further. So you have these different areas here, and then so maybe over here you're building a base of something, and over here it's going to be made into a TV. Over here it's going to be made into a monitor, etc. Once you make that item is then packaged and set up for receiving, and it goes to different areas, or you can store that item over here. Right? That's a factory. Cells are a lot like factories, only cells are such good factories in that they actually, one of their main product is themselves. And so actually, so this is another product. This is in German. And so this is showing you, this is an ice cream factory. It's showing you all the steps that are going on of what's going on with an ice cream factory. I'll get on it. But there's a number of things that are done in a factory. We tend to not see them, but a factory has to get built. It has to be made. We have to take all the raw materials and build a factory. The great thing about cells is they're such a good factory is that they can make themselves. And again, really good factories in the United States, a lot of times you're kind of building yourselves. You will have a unit in that factory that's responsible for maintaining and building itself. And so what we do at a factory is factories build themselves. Factories make decisions, right? What do you make? How much of what do you make? What materials do you have that you can make? Factories have instructions to make both the factory and the raw materials. Factories will collect raw materials. Factories will maintain their internal conditions, right? If it gets too hot, that may affect the factory. If the factory's underwater, that may affect it. And so cells, too, can maintain their internal conditions. And one of the big things is factories make a product. That's something that's used. And again, cells, for the most part, most cells produce a product, right? So if you're a secretory cell inside of a human being producing some type of glandular material, you will actually make something. So lots of cells make materials that are exported outside the cell. When it is exported, or even when it's moved inside the cell, materials are packaged for use. We see product delivery, and then any others. Can anyone think, are there any other things that factories do that kind of cells do? So I'm trying to kind of get you to think about, I'm getting you to think about cells by kind of thinking about something different, a factory.
Hello.
Karamoko, yes.
So, well, repackaging?
Kind of repackaging, so you're taking materials that you already have and you're packaging them up in a different format. Sometimes it's raw materials, right? And so you may have somebody that gets something that's already made, like a car. You buy a car and then you put your own brand on it and then you sell it to somebody else.
Mm-hmm.
I've seen that done sometimes.
Distribution.
Distribution. Yeah. So you, again, a cell is going to, the cell is constantly making stuff, but again, you need to distribute the materials in the cell to where it needs to go. And a lot of times if a cell, especially if it's part of a colony, it has to, if it's making something for export, it needs to be able to make sure that's distributed to that piece. That's true.
I was thinking of, well, not a typical activity, but what the factory is like made of and how it's like all the iron or support beams or the
Yeah. So you're
so
gonna,
I would think about cytoskeleton.
yeah. So you're gonna build the factory. So you need to build the parts. So you need to get the metal to make the supports on the factories. And again, part of that you need to have the plans that allow you to kind of make the factory.
Professor, do they have also like quality control?
Oh, you bet they do.
Conveyor belts,
You bet they do.
like stuff flowing through to make sure.
So you have these little microtubules that will actually move stuff. It used to be we thought things would just move down a concentration gradient, but we're seeing that there's actually cells can move. When a cell makes something and it's in the cytoplasm, it could actually move it back into the nucleus by using these little microtubules.
What about like creating waste or dealing with waste? Actually,
Dealing with the waste is a big one, yes. And so cells will have that as well. Cells do a lot of recycling, right? You want to recycle it. So if a cell has a protein that's broken, what it will do is it will actually use vesicles, and it will break down that protein into the amino acids. And then those amino acids can be used by the cell to make something. So the great thing about life, it's a very good teacher for us, or a very good example for us, in that everything that a cell creates for the most part is recyclable. There's always examples of different things. And so wouldn't it be really neat if we were to make stuff? So we make, how many of you are seeing these masks outside? Wouldn't it be great to have these masks so that, and actually these are pretty recyclable. The ones that are made out of cellulose are recyclable. The ones that are made out of polypropylene are a little bit harder to recycle. But wouldn't it be great after you did your mask that you ate it? Made out of cotton candy, and you just ate it, and it was gone. And it'd be totally recyclable. Okay. So what I'm going to do is I'm going to give you guys access to a Google document. Let me see if I can find that Google document. All right, so let's just go back. So if we go back to, I don't have it on here, but I should have it under assignments. And I'll put it on there. So under assignments, we should have microscope validation that's coming up. Cells are like factories. So if you click on cells are like factories.
Every piece of the cell is used as well.
Yes. And of course, I don't have that in here. So what I'm going to do is I need to make that. Oh, wow, somebody's near. All right. So I have something called cells are like factories.
Huh?
All right. Okay, so in actuality, I did this yesterday. So what I was hoping to do is kind of get you guys into groups and have you kind of discuss what a cell is.
Is this a relatively quick assignment, or is it going to take a little time?
Well, you know what? It's sometimes what I think is a quick assignment takes you guys a little bit longer.
The only reason I asked you that because I see that it's due tomorrow, and I'm not done with class tonight till 10 o'clock. So I don't know if we could kind of push that more than a day.
I'm going to give you about a half hour, and
okay. Because I have back-to-back classes. So as soon as I'm done with you, I have other classes.
Okay. All right. So what I'm going to have you guys do. So this is the assignment. In class, we discussed how cells are like factories. They can build themselves, make decisions, store and present instructions, etc., package notes. Among your group, share a Google document where you expound on this idea with either a drawing, figure, slides, movie, paragraph, or other multimedia materials. Make sure that you show and name the parts of the cell and tell what they do in terms of a factory. Make sure that each member of the group name is on the document. Each member can share the document with your instructor or share the URL. So what I want you to do is to do this, share a Google document among yourselves, and then tell me, give the parts, tell me what the parts are you can do. It's kind of open for you. You have about a half hour to do it, okay?
Okay.
I'm going to flip you into groups of 3 soon.
You're going to send a link, right?
No, there's no link.
Oh, okay, okay.
That's an announcement. So you're just going to make a document. So have somebody in your group make the document, share it amongst yourselves, and then kind of put the materials in the document. Really, I don't want you to take more than a half hour on it. I want you to start it. So you will get points for kind of starting this up.
Okay.
Has everyone got it? Does anyone have any questions on that?
We're good.
And are you going to drop the link for the stuff we can relate it to in the chat?
It's in there, and yes, I'll try and drop it in the chat for you. Breakout rooms.
But we need access to the document before you throw us in the breakout room.
No, you guys are going to make your own document. So one person in your group is going to make a document. You're going to share it with other people in the group, give them editing rights. Okay. And then you're going to, it's what I call a swarm document. You work on that document and you answer those questions. So basically you can take a cell and explain to me why it's like a factory, what the parts are. So name the part, take a cell, name some of the parts of the cells and tell me what they do and tell me why what they're doing is like a part of a factory. Okay?
Nick?
Yes, ma'am.
How many parts in general do you want us to describe? I mean,
as many as you can given the time constraints that you have. Okay. I would not be happy if you did one. But again, because you have a half hour, you should be able to do most.
So like 10 parts, or are you thinking more than 10? Just a sort of a ballpark number.
I'm going to say most. Leave it open.
Okay. All right. Thank you.
All right. I'm creating the room, so you're going to go there right now.
Okay. Thanks.
Okay.
Hi, how's it going?
Hi. Raphael, did you make it into a room or
Yes, I did. Thank you.
do you have a question for me or something? Or
I'm trying to talk to my group. Oh, sorry. So I'm looking on two accounts. I'm sorry.
okay. Okay, popping in. Does somebody make the document? You're sharing it?
Yeah, I'm working on it right now.
Okay. Split it up. Good luck. If you have any questions. How you guys doing this, Nick? You guys okay?
Yeah, we're good. I think so, yeah.
Okay. All right. Just checking. Catch you later.
Okay.
This idea with either drawing, speaker, slides, movie, paragraph.
Yeah.
Sorry, guys. You ended up with just like a group of 2, but that's okay. I'll be a little bit easier on you. Just try and do what you can.
Okay.
Any questions or anything?
All right.
No, we're good, Doc. Thanks.
All right. About 5 minutes before the end of class, we'll pop out together.
Cool.
All right. Thanks.
Yeah.
All right. Sorry, guys. Cecilia, how come you always end up with a group of 2? I don't know why. But,
I don't know either.
but just try as much as you can and stuff like that. You guys, you'll get a document and kind of send it off to me, and whatever you have, I'll take.
Cool.
But I just want to know is you have a group of 2. Any questions or anything?
No.
No, I think we're good.
Okay. I will see you 5 minutes before the end of class.
Sounds good. Thank you.
Hello.
Hi, guys. Sorry, most groups have 3. You guys have 2. I'm sorry about that. So, if you do a little bit less, that's okay. You guys have a document that you're sharing and you're trying to discuss and describe the cell as a factory and describe.
Yeah.
Okay. Any questions or anything for me?
No, I think we're okay.
Okay.
Yeah, I think we're good. Thank you.
Great. I'll call you back to class about 5 minutes before the end of class, and we'll have a discussion. Okay. Bye now.
Okay.
All right, Andre, how'd it go?
I think we did pretty well. I mean, we looked it over, and hopefully we're not missing too much, but I think we got the main components of the cell.
Okay. That's good. I was just, I'm just trying to get you under adverse conditions to kind of think about these things and stuff. So it's practice for the exam.
Yeah, definitely. Wait,
So
you didn't care whether it was a plant or animal cell, right?
At this point, no. Plant, animal, or prokaryotic. And as long as you got some of them, and as long as some of them are right, I'll
Okay.
take it. All right. How are people doing?
Round one.
Does anyone have any questions, comments, concerns?
No, not yet.
So that was meant to be kind of like an example of possibly an exam question that you would get. You're going to get a very little bit of time. You're going to have to think on your feet, and that's okay. Whatever you put down is what you put down. I'll take a look at it and see.
How many do you want? We covered about 10 so far.
10's great. I was thinking I'd be happy if you did 3.
Oh, we're going to keep going. We're just going to name every part of the cell and label everything if that's what you want.
Okay, but I want you to compare how it is like a factory. Label stuff, that's great, but it's not. What I want to know is kind of how is it like a factory? What is it making? What's it involved with?
We've got all of that.
Okay. All right. So you can keep on going, and I'm probably not going to see them for a while. These are all on Google Documents, so I can take a look at them.
Sure. We'll post as many as we can, and then we'll have the breakdown of everything and how they relate to one another.
Okay.
Okay.
Anyone else? Anyone else have any questions, comments, concerns? Okay, so that's basically about it. We have an exam coming up in 1 week. There's a paper that's out there that's available for you to create your own study guide. I suggest that you work in groups of 2 or 3. You've been meeting people in the class, kind of get together and share what your answers are and discuss what your things are for the study guide. There are discussion boards that are up. Please participate in the discussion boards. I will be monitoring them. It looks like things are supposed to clear up in terms of the smoke for Thursday. So I think Wednesday is still a possibility that it may be canceled, but I'll let you know, or you'll get an email me for either from me or from the district. But in the meantime, I've also posted up an at-home lab that you can do. All right, I will, you got 2 minutes to go off and have fun before your guys' next class. All right, I'll hang out for a little bit. If you wanna talk or do something during my office hours, that would be great. Catch you later. Sabrina, Ryan, oh, so people are popping in. Thank you. Katrina, thank you. All right, thank you. Yeah, make sure that you put something in the chat. I will record this and hopefully put it up soon. I'll also that's one thing I need to do first is fix the lecture for chapter 3.
Professor, your email address is ncap@mysmccd something, right?
No, it's just CAP, K-A-P-P at
Okay.
smccd.edu.
Thank you.
The one that works best for Google Drive is nick.cap@gmail.com.
Oh.
Sorry, and we just submit on the assignment. You just submit the link on the assignment.
Yeah, so what you're going to do is to go in our assignment, and then you submit that link, and then you can give me, everyone in your group could do it, so that way I kind of see the same thing. But if you put your name in there, so that way I can take a look at it.
Okay.
And what I do is I tend to grade it. But that way, if everyone puts it in, if everyone puts in the link, then it's done, rather than, oh, Bob was supposed to do it, and he flaked.
Okay. Professor, you said it's nick.cap@gmail, right?
Yes.
Okay,
let me
great.
put it in the chat.
Thank you.
Professor.
Yes, sir. Alec.
I just want to like check in with the homework assignment. We did slides, and basically what we did, just each slide we just did like an example, a specific example for this one. So like I'll just give you like one of the slides. So like taking out waste, and then we did like in the factory there's like garbage disposers are in charge of taking waste.
Okay.
And so in a cell it has a lysosome like who charge of digestion and waste removal, and that's pretty much it. And then we're just gonna move on to the next slide.
Yeah,
Okay.
That's it. But the idea is, rather than you drawing me a picture of where a lysosome is and saying lysosome is, does waste, I want you to kind of do that comparison, right?
Yeah.
Yeah.
Yeah, like our title is pretty much like what they like both do. They both do waste removal, and then we did what do they have in the factory and what do they have in the cell that's similar to waste removal, and kind of like that.
No, that's great.
Okay.
So Dr. Campbell, I had a quick question for you. Can I just, I want to make sure that I'm doing this right. So like, for example, when I talk about the nucleus, I talk about how it's the executive department or the headquarters of the cell, right? And I basically say it runs
like a factory, it's the headquarters of the cell. Yeah, I'll take that.
Okay, then I say it runs, and then I go in detail and I say it runs the cell factory and controls all cell activity. It determines what proteins are to be made and stored, and all the plans for any proteins that the cell currently makes or has made in the past.
Yeah.
Okay.
That would be good.
Okay, because
as long as it's close. I mean, there's other issues that determine what proteins are made and when and stuff like that, but ultimately it does deal with the nucleus.
Okay, so do you want it kind of set up like that, basically telling you what the nucleus does and then how it operates in a factory setting, basically?
Yeah.
Okay.
Yeah.
Good. Okay.
Well, that's fine. And I'm not going to split hairs on, it's not like this and stuff like that. As long as it's close, that's fine. That's what I'm looking for.
Okay. Great. Thank you. Talk to you later.
Okay.
Thanks, Professor. Bye.
All right. No problem. Bye now. Hey, Gabriella, how's it going?
It's good. Just working on the assignment still.
Professor Cap, quick question. Since people who have lab on Wednesdays, do we go to lab today?
I mean, if you want to do the lab, you can. I mean, because that lab, you're essentially going to need to do that lab that I have, the assignment that I sent out today. You're going to need to do that, but you don't need to do it. I gave everyone like 2 weeks to do it. So
yeah, I meant to say I'm in Oliver's lab session. So should I log on at 1:10?
For today, no, because if you're in a Wednesday, you don't need to log in for today.
Okay.
What you want to do is say, hey, Oliver, I was going to do this lab or whatever. We don't know.
Okay.
So, for the most part, I don't know what's going on Wednesday.
Okay.
If you want to this is a lab that you have to do anyways. If you want to try and do it.
Okay.
But I would say wait till he gives his instructions.
Perfect. I'll just pop in then at 1:10 just to be.
Okay.
Okay, great. Thank you so much.
Mm-hmm. No problem.
Hi. I just wanted to give you or let you know that I submitted my valid checklist late.
For the gram stain. You know,
I knew that was on Wednesday, and I got your email yesterday about submitting it on Saturday. I put a little personal note, but on my submission, just
yeah, it's fine. And this is
is rough, but I submitted it, so I hope that you can take a look at it for Wednesday.
Yeah, I will take a look at them because again, we're not doing the lab. That slowed it down and stuff like that. And again, on all this stuff, it's okay to hand it in late. Sometimes you'll get a few points, but you're not going to flunk because you were hours late putting something in. So that's not my intent. Thank you for trying to keep up and trying to do stuff. So I appreciate that.
Okay, see you Wednesday.
Right.
Hey,
right.
Cecilia.
Yeah. Oh, you want to help me?
Yeah. Can I just show you now? Is that okay, Professor? Doctor?
Sure. What are you going to do? I shared the screen. You should be able to share your screen if you want.
Okay, let me just prepare it. let's see. Can you see it?
Mm-hmm. Yeah.
Okay, so I'm just at Skyline. I went to the website, and then I just go here. And then I believe, let's
Wait,
see.
where did you go? Hold on, the faces were in the way.
Okay, so yeah, I'm just at the Skyline College.
Okay.
And then I go here, I go to where it says A through Z.
Okay.
Like S for STEM Center.
Mm-hmm.
And it'll take me through all the things that start with S that's on the website.
Mm-hmm.
And then go STEM Center. And then down here, it says join the Discord server. So it's something that you're going to have to download. So, just make sure you have enough space for it. But basically, I already have it, so it might just direct me to it.
Okay, let me try that because I think that I've signed up for it already. So hold on 1 second. Let me just try and see if I can at least get to that step.
Mm-hmm.
Okay, so I think I made my account. Okay, well, I'll follow you and then I'll figure out if I can log in.
Yeah.
Okay.
So I guess continue to Discord. I guess you can do it online or via the thing you download, the server that you download. And basically, I don't know if can I share my my background? Oh, I guess this is what it looks like. Yeah. So I'm already logged in. So these are mine, but let's see. This one is the one we're on. And usually before going into the whole thing, you'll get a notification from, I think his name is Brian. It usually shows on this side, everyone. But it's just to kind of get your information as a student.
Mhm.
So your ID number and your class that you're registered for, and what teacher, I think. But yeah, just to make sure. And then once you submit that, they'll give you more access. But as you can see on the left-hand side, there's math tutors, there's biology tutors. So for us, this would be ours. And so there is a tutor on here. I think this is Benjamin Lamb. But usually he'll say, okay, I'm available to tutor till 4, or from 3 to 4, or 2 to 3, or till 5. And so he'll post here randomly. Also, the other professor, Professor Oliver, I don't know how to say his last name, but yeah, he holds his office hours in here.
Okay.
But also, at the very beginning, you have this little section called welcome, the general info, and they'll have screenshots of how to maneuver around the website. So
oh, that's helpful. Okay.
Yeah, so you see here, it shows you the members, and oftentimes it'll be labeled let's see, the service coordinator, which is Brian, and then the leaders, so the tutors. And it'll even show you students too.
Oh, cool.
And then here's everyone who, or not everyone, but the classes and the different channels that they have.
And so you can just click on these classrooms and then join Zooms, a group Zoom for tutoring?
Yeah, so they'll often have a link. So this was a long time ago. But yeah, you just click on the Zoom link and then you can go chat in with them.
Oh, cool. Okay.
Yeah, look, Amy wrote something.
Oh, got it. Okay.
Yeah,
So
so you
this
can
is
just
a lot on the back
what's up,
end.
guys, right? But
Okay.
Yeah.
And then, Dr. Cap, do you know, will you be holding, do you use this platform?
Yeah, I monitor the platform. So I'm in there. I try and do stuff. I don't necessarily, because I usually do kind of before and after class for my office hours.
Yeah.
But I can meet people in this.
Do you know if there will be like a study group before the exam that we could join, or can we create one?
Yeah, let's create one. I'll try and do that. I'll try and do that for Wednesday. Gabrielle, is it okay if I post this? I'll make a copy of this and post this for other people to look at. I think you did a great job.
Oh yeah, it's recorded, huh? Yeah.
Yeah.
Okay. Yeah. Sure. Do you have any questions?
No, but thank you, because now I can just like go in and explore, and if I have questions, I'll reach out. But thank you for just, you know, I don't know, I feel like it's not super user friendly, the navigation of it, but I think it would be super helpful to use.
Yeah, I think once you use it, it gets friendlier as you use it. I kind of am on and monitoring it. So if you ask me a question or something, a lot of times it will ping me, and I can go on it from there.
Yeah, and
like
you can see on the right side, it has like all the professors. So like right here, Dr. Cap. So
Yeah.
Cool. Okay.
So see, that's a little green light in there. So it says I'm online and I'm doing it. So.
Oh, got it.
So.
I feel like for some of us who aren't like super technologically inclined, there's this like learning curve. Like you're learning the course material, but you're also learning how to do it from home.
yeah, my 13-year-old cousin, like before I started this class, like was like, Gabby, do you have Discord? Like, let's chat there. I was like, no, I don't. And so he like walked me through all of this before the class even started. So I was like super happy.
That's so cool. Yeah, we have I have a 7-year-old, and so he'll be like, oh, mommy, you have to do this on the Zoom, you know, and like here's the chat and you can write on the screen. And like,
oh, that's cute.
You better not be doing that in your class. You better be paying attention. All right. Well, thank you, Gabriella. I really appreciate it.
Yeah,
thanks for the time, Dr. Cap.
Okay. All right. Catch you guys later. We're done? Okay.
Bye.